Okay, let's take a look at the enzymes.酶. Enzymes are proteins that serve as catalysts for reactions at the physiological conditions, including constant temperature, pressure, osmotic pressure, and pH. Some enzymes require coenzymes, such as metal ions and small molecules for enzymatic functions. Enzyme activities are tightly regulated. 酶是在生理条件下，比如恒温、恒定压力、恒定渗透压或恒定 pH 值，充当反应催化剂的蛋白质。一些酶需要辅酶，例如金属离子和小分子才能发挥酶促功能。酶活性在生理条件下受到严格调控。Next, let's take a look at type of enzymes. Some enzymes are classified into the following types. 酶的类型，一些酶呢分为以下类型。The first one is called oxidoreductases. Oxidoreductases catalyze oxidation reduction reactions. 氧化还原酶催化氧化还原反应。This is the example. This is called this enzyme is lactate dehydrogenase. The product, the reactant is private and NADH. Then they form a lactate and NAD plus. The change is here. You see this carbonyl group and become a hydroxyl group. There's a hydrogen here transferred here, and the carbonyl oxygen become a hydroxyl here. Next is the transferases. Transferases catalyze the transfer of a group from one molecule to another. 转移酶催化从一个分子到另一个分子的集团转移。This is an example. This is called alanine transfer aminonase. Alanine transfer aminonase. Basically, this enzyme transfer amino group of alanine to alpha ketone glutarate, become pyruvate and glutamate. This is the reaction of regarding the amino acid conversion. The alanine, this one amino acid, is converted to another amino acid, is a glutamate. Next is called hydrolysis. Hydrolysis catalyzes hydrolysis. Cleavage the bonds with water. 水解酶催化水解，使用水非烃裂解化学键。This is the example. This is lipase. This is a triglycerol. This is 3 water, and then this ester bond and 3 here cleaved and become glycerol and 3 fatty acids. Number four, Isomers catalyze conversion of one isomer to another. 异构酶催化一种异构体向另一种的转化。The example is triose phosphate isomerase. The reactant is called dihydroxyl acetate phosphate. This is dihydroxyl acetate. This one hydroxyl group. This is supposed to another one here, and this one is used to link the phosphate group. Then the product is called glyceraldehyde 3-phosphate. The change is this area, you can see. Those two molecules are isomers. They have the same formula, but the structure is different. And you can see here in this molecule, this carbonyl group become a hydroxyl group, and the hydroxyl here become a carbonyl group, and these two the isomers. Number 5 called Lysoses. Lysoses catalyze the addition of a molecule to a double bond and or eliminate a molecule to give a double bond. 裂解酶催化添加一个分子到双键，或者去除一个分子以产生双键。This is the example here. This enzyme called fumarase. This is the fumarase. This enzyme. Okay, they have the double bond here, and then you can the water. Water has two groups. One is hydrogen, another hydroxyl group. It's water, two groups, and then these two groups added to this fumarase. The double bond, one double bond breaks, and one hydrogen added on this carbon. The hydroxyl group added on this carbon, then they become Isomers. You see this hydrogen is this hydrogen. The hydroxyl group is this hydroxyl group here. The last type is called Largesses. Largesses catalyze joining two molecules into one molecule by forming a covalent bond or joining two ends of a polymeric chain to form a ring. 连接酶来催化，通过形成共价键将两个分子连接成一个分子，或者连接聚合物链的两端形成一个环。The example is pyruvate and carbon dioxide. They link together to form oxaloacetate. And you see this pyruvate has 3 carbon here, and oxaloacetate has 4 carbons, 1, 2, 3, 4, because the additional carbon in this molecule comes from the carbon dioxide. This reaction requires energy. ATP is high energy molecules, and they provide energy. The ATP become in this reaction, ATP become ADP and phosphate. And the final part, the last part, let's take a look at naming, how to name an enzyme, 给酶命名. In general, the name of an enzyme includes 2 parts. 一般来说呢，酶的名称包括2部分. The first part identifies the substrate the enzyme works on. 第一部分确定酶作用的体物. The second part identify the type of the reaction catalyzed. 第二部分确定催化反应的类型. Example: succinate dehydrogenase catalyzes the removal of 2 hydrogen atoms from the substrate acetate assassinate. Pyruvate decarboxylase catalyze the loss of carbon dioxide from the substrate pyruvate. 四例，琥珀酸脱氢酶催化从低物琥珀酸中取出2个氢原子，丙酮酸脱酸酶催化从低物丙酮酸中取出1个2氧化碳分子。Some enzyme names are derived from the substrate by changing the ending to ASE. For example, lactase catalyze hydrolysis lactose. 一些酶的名称是从低物衍生而来的，将结尾更改为 ASE。例如lactase 的乳糖酶催化lactose的水解 ，lactose is 乳糖。